All right, now I would like to welcome to the stage Professor of Neurochemistry and ADRC Biomarker Core co-leader, Dr. Henrik Zetterberg, to introduce our next featured speaker.
Thank you very much, Barb. It is my great pleasure to be here, and I will give a short introduction because we are cutting into Rema's time here. But you are in for a treat now, because you will listen to actually the world's leading expert on clinical trial design in brain diseases. And Rema has designed studies in stroke, you have traumatic brain injury, it's MCI and Alzheimer's disease. And the way, when you speak to Rema about her research, you will find out that she, if you have an Excel sheet with a lot of data points, Rema knows, she thinks about each individual person behind these data points. And she's a mathematician by training, and that sounds scary, but she's super nice. And you can ask her anything. She will be around for perhaps 15, 20 minutes. But if you have additional questions and get really interested in Rema's research, you can also listen to Nate's podcast, because this morning, Rema was grilled in this podcast. It was supposed to take 10 minutes, but you spoke for 30 minutes, I heard now. But Nate is taking out the really good things and perhaps some fun things too. Keeping in. Not taking out. It was the opposite. That happens to me sometimes. Now I will actually just give you the stage, Rema. Welcome, and so happy you are here.
Thank you, Henrik. That was awesome. Hi, everybody. Just wanted to thank Dr. Bendlin, Dr. Stana for inviting me here. It's just been an awesome trip, meeting all of you yesterday and then being here today and listening to all of this research. Just a little bit about me, and to at least get an idea of the perspective that I bring to today's talk. I'm a trained biostatistician, and I've been a trialist for several decades now. So I call myself a clinical trials biostatistician. And about a couple of years ago, in the interest to making our trials more inclusive, I got more and more interested in recruitment and retention science. So today's talk is going to sort of bring all of those perspectives together in a way to at least give you a flavor for what we have been doing in AD clinical trials research in the last several decades. And thinking about how we can accelerate the next generation of trials through, in my opinion, inclusion as well as workforce development. Oh, this is what you told me. I have to do both. Okay, that work? All right, and I want to begin my disclosures, which is my current research support. I have the pleasure of being part of several research teams, all of us working jointly towards trying to figure out ways to find better treatments for the disease and better ways to measure outcomes and design better trials. So I'm going to do it in 3 segments, and I want to start by putting this whole conversation into give some context to the conversation and talk about what do we know around this work at this point in time. I'm going to just talk about some aspects of it, and where is the need that we have to address as we move forward in the field. And I want to begin with this picture from the annual paper that Dr. Jeff Cummings puts together around the drug development pipeline. And I'm going to talk primarily around drug development and pharmacological trials, but a lot of what I'm talking about applies to non-pharm trials or any other clinical trials. And this is the 2023 pipeline. And the key things that I wanted to get across was this, that we have 187 trials right now in drug development, assessing 141 drugs. And what this means is we need over 57,000 volunteer participants to complete these trials. And remember, this is just industry trials. NIA has funded almost 400 plus other trials that span pharmacological trials, non-pharm, caregiver trials, and other types of trials. So we're talking about a field that is really active in drug development and in therapeutic development, with the need to enroll volunteers to participate in these trials in really large numbers. And I'm going to keep switching gears here, so bear with me here. Hopefully at the end, this whole story comes together. The second thing that I wanted to talk a little bit about is, for those of you who are not familiar with randomized controlled trials, why is the design impacting the trial recruitment? How does the study design actually impact trial participation? And I want to talk through some aspects of the clinical trial design, especially in pharmacological trials, which I believe are some of the hardest trials to recruit participants to. So you're talking about, in the spectrum of ease of participation, recruitment into trials, this sort of falls on one extreme of that space. And the reason is that in a trial, irrespective of what you're studying, the primary research question of interest is the intervention safe, and does the treatment work? You're only interested in the treatment effect, and you only have sufficient power to answer that question with the entire sample size. Everything else are subgroup analysis, secondary analysis, exploratory analysis. You have one research question. It's about your treatment effect and evaluating the biological effect of your drug that you are studying. And because of that, because you haven't actually demonstrated that effectiveness or efficacy, the inclusion and exclusion criteria for trials tends to be very narrow, with a focus on individuals with the disease being studied. And the intent is to minimize likelihood of harm. So in other words, if you're looking at an anti-amyloid therapy, you're looking for individuals who have sufficient levels of amyloid in their brain so that you can evaluate whether the particular intervention actually slows that accumulation. So you don't want individuals who don't have sufficient amyloid in the brain. And the reason I'm saying this is because of some of the rest of the data that I'm going to show you. Because of that, the intervention delivery tends to be very strict. You don't have deviations that are permitted in most of these pharmacological trials. Randomization and blinding is an integral part of these trial designs. Individuals are, by a flip of a coin in most cases, randomized to receiving an active, but the treatment or placebo. That's something that needs to be talked to in terms of participants. Participants have to be willing to be randomized to getting no active intervention. And in addition, these are all double-blind trials. In other words, you don't know what you're getting. So you have to trust your research team that they are going to take care of you in a manner that is in your best interest. And all of this has a huge part to play in participation in trials. And because of all of this, because you haven't yet evaluated safety and efficacy, the safety oversight and the regulatory requirements for trials tend to be very, very high. And all this is really to get across a point that I want to make, which is there's been a lot of conversation in the field around the need for what is called representative trials. And I just wanted to get that point across that in general, clinical trials or explanatory trials, trials where you're still trying to figure out safety and efficacy, are focusing on internal validity. That's what you're trying to do. You're maximizing internal validity. In other words, you want to be sure that when you get the results, it is valid. What pragmatic trials do, as opposed to explanatory trials, is maximize external validity. And it's a nuance, but it's a very important nuance. So as a field, as a team, we are moving away from representation to using the word inclusion when we think about our trials. We want inclusive trials, recognizing that representation is much further away just by the issue of our trial design. The next several slides, each slide has a lot of content, and each slide can be a talk in by itself, but I just again wanted to get across some of the things that we are hearing and learning from the work that we're doing. This is some data from 3 different sources. The top left is data from a paper from Ritchie et al. From last year that shows you, that talks about the average number of participants randomized per site per month. Remember, I told you in the beginning, we need over 51,000 participants. Well, right now, we have sites randomizing less than 1 participant per site per month. And that is on a good trial. In most cases, it's about 0.3%, 3. So you have 3 months to randomize each participant. If on the bottom, no, the top, the left bottom is the results of a systematic review. And this looks at race and ethnicity, and it looks at who is participating in these trials. It's a global systematic review, and so you can see that there is the skewness in the distribution of race and ethnicity demographics in trials. And the trials in that percentage of numbers on an average from Asian and Hispanic communities are driven by the fact that many of these trials have sites in Asia or Latin America. So if you look at this in the US context, the numbers of Asian as well as Hispanic participants are much, much lower. On the top right is data from the Alzheimer's Association's facts and figures from a couple of years ago that really the bottom line there is it's looking, they asked questions around why individuals would choose or not to choose to be part of trials. And you can see that their reasons differ depending on which group, what your experience with the medical system has been. And this is important for us to know because as we think about recruitment strategies, it's important to know that one size does not fit all. It does not fit all communities. It does not fit all regions of the country. So it's something to keep in mind as we think about recruitment and retention approaches and strategies. Oh, and the other point I wanted to make was that you don't see data on any other dimensions of underrepresentation. We focus on race and ethnicity, but there are several other dimensions of underrepresentation in our trials. The next slide, I think others in this room have much more knowledge about this, but this is data that we are starting to evaluate in terms of who is making it through our screening process for our preclinical and prodromal trials. And there's, again, a lot of data here. I'm just going to walk through this slide. On the top left is the data from the A4 trial. And A4 was the first preclinical trial, AD trial, looking at solanezumab, just concluded. And the data for this, the full data set, will be released in a couple of months. But this was work that we did a couple of years ago once screening was complete. And what we saw was that there is a lower likelihood of individuals from certain racial and ethnic groups meeting the amyloid eligibility criteria that was required to be eligible for the A4 trial. Center bottom, you see the same data, similar trends, but this is in early AD trials. This is looking at a meta-analysis across 4 pharma trials, AD trials led by Josh Grill and our team. And this is looking at also relative amyloid PET positivity rates. And you again see this differential eligibility across race and ethnic groups. On the top right is data that has recently been published. It was just published a couple of days ago by Dr. Doris Molina-Henry from our team. And this has 2 plots. Wanted to take a minute to talk about. On the left, so the first 2, A4 and early AD, were based on PET. On the top right is now we've moved for the AHEAD study into plasma. And in the early part of the AHEAD study, we used plasma A-beta 42/40 ratio to predict amyloid positivity. Now we use p-tau217. So this data is using the 42/40 ratio. And what you can see at the top left is at the point of the first visit, way before cognition, way before PET, you see this differential eligibility rates using plasma markers, very similar to what you see using PET. Once you adjust for that, once you make it past the plasma criteria, after cognition, once you get to PET, those differences no longer exist. So there is this data accumulating around the levels of amyloid burden across racial and ethnic groups. And there's a lot of work being done in this space by many scientists in our teams and across the country. So there are these differences in biomarker eligibility rates, which are enrollment criteria for anti-amyloid trials, and it skews the race and ethnic distribution of the randomized samples in these trials. On the positives end, for those of us who've been working in drug development for two decades, this last couple of years have been an exciting time. And I hope I can get that excitement across to all of you. We have our first fully approved amyloid lowering drug, a disease modifying drug. It's not just about helping people with disease, it actually lowers the biology of the disease. It's a really exciting time in the field. This was given traditional approval by the FDA, and it is being covered by Medicare, at least here in the United States. It's covered, I believe, in Japan. The European Union is still evaluating this drug. But the reason this is important is for those of us working in AD trials, it impacts the design of these trials, it impacts the conduct of these trials, and also impacts the management of these trials, because now you have individuals coming into your trials who are in that preclinical and early AD stage who might be on a drug that actually reduces the amyloid levels. These individuals have higher risk of ARIA. So safety management becomes an issue, and study management becomes an issue. So these are all things that as a field, we now have to be prepared for as we think about the next phase of drug development. Connecting all of this together and why inclusivity becomes so important is because finally, a few years ago, the FDA guidance has come out, which about enhancing the diversity of clinical trials participants. This guidance was really an important part in the field to help the public-private sector in particular get on board with this. And so now both in the public and the private sector, representation, inclusion, all of the terminologies is being used as we try to develop and establish inclusive trials. And the recommendations are broadening the eligibility criteria, designing trials that are accessible, and talk a little bit about some of the work we're doing in that space, adopting enrichment strategies, and then ways in which to enhance inclusiveness. I was gratified to see the word inclusiveness rather than representation in the FDA framework, which is around enrollment, diverse participants and staffs, and that gets to the education and training workforce development part of why of the work that we're doing. General engagement of multiple sources. I'll talk about 1 and 2 a little bit today, but there are other aspects around cultural competency, geography, and trial efficiency that we're also working with. And finally, clinical trials workforce. It's probably the most important and the least discussed aspect of the work that we do. Everybody's really excited about trials, but you can't do trials without a trained workforce. Without trained workforces at the principal investigator or the leadership level, and equally important, the research teams who are actually managing the conduct of these trials. So I want to talk a little bit about that today as well. So on the left are some of the challenges that all of us working in trials have in academia as principal investigators or co-investigators. We all know that there is underrepresentation of minoritized groups and women at all levels of academic medicine and industry. This was a bullet point that was from several years ago. Things are changing slightly, but we're still not there yet. There is a need for a diverse, well-trained ADRD clinical trials leadership that is reflective of a specialized discipline with a focus on team science. Clinical trials are never done by one person. It's never done by one group. It's really done as a part of a team science approach. And it goes counter to how we are evaluated in academia, where the PI is the only one who gets the credit for this entire program. And so it's a cultural shift that all of us in the trial space have been working on for several years. There has been inadequate institutional support for all of us wanting to work in trials. And this is again driven by, think about those of us who have been part of trials programs, but hit a roadblock when our promotion comes up or our tenure comes up and our academic files are being reviewed. Participation in trials, involvement in trials is not given that level of importance as it probably should be. And there is a lot of work that is being done to try and change that approach. And then the lack of community physician involvement. The research teams still work a little bit in silos and are not connected with the community physicians, which is where the majority of the study participants actually interact with. That's where most of the trust is at the community level. And on the right is a recent paper, actually a couple of months ago, from Friel et al., that talks about the impact on the field on research professionals. And this is a huge element of the work that we do. And there is this, the data that shows that there is a huge shortage of clinical research workforce. Oops, somebody tell me, I'm sorry, right up there. I was so busy talking, I forgot about changing the clicker. And I was looking at all of you all, not one person looked like, okay. So the clinical trials workforce shortage, and this is happening with individuals who actually are more experienced. And as a result, the current workforce is overburdened. There's been a high resignation rate for professionals with 5 to 10 years of experience, and a high turnover rate between 35 and 61% at our sites. And we see it at the coordinating center as we talk to our sites. And this has a huge impact on the ability of the teams to conduct trials, to see participants, to conduct trials, and to retain participants in trials. So the impact of this is huge in the field. So with that, I'm sort of going to shift gears. So that's really where we are right now. Exciting times, drug treatments approved, which means the next stage of treatment development is ongoing. We've got farm treatments, non-farm, combination therapies. Exciting time. We've got biomarker development, super exciting time in that space, where we are now able to use biomarkers instead of PET scans, blood, to be able to screen participants through the system. We're moving into the community. However, we don't have the workforce in place. We don't have the academic, in an academic environment, we don't have the credit that is given into this, into being a trialist. And we have other challenges that we have to work with, including inclusivity in our trial. So what are we doing about it? So I want to shift gears to talk about the Alzheimer's Clinical Trials Consortium. University of Wisconsin is a member of the consortium, Dr. Carlson here, and the team. But I want to talk a little bit about the consortium. I want to set it up in the context of the work that we just talked about. So the Alzheimer's Clinical Trials Consortium is the federally funded, NIA-funded national consortium conducting trials in AD. It's led by 3 investigators around the country: Paul Azin from USC, Ron Peterson from Mayo Clinic, Rochester, and Risa Sperling from Harvard. And the 3 of them, along with Laurie Ryan from the NIA, form the leadership team of the consortium. The role of the consortium, it's an infrastructure grant, and it is to provide an optimal infrastructure, utilize centralized resources and shared expertise, all with the aim of accelerating the development of effective treatments, both for AD as well as for ADRD. It's a consortium that's designed to conduct both phase 2 and phase 3 trials, not phase 1. We tend to stay with 2 and 3. All stages of AD and AD-related dementias, symptomatic and disease modifying. It includes behavior, non-pharmacological, as well as pharmacological interventions, and it encourages public-private partnerships, such as the AHEAD study, which is a public-private partnership between the NIH and ACTC and ASI. There is funding for central infrastructure in the ACTC, and what this means is, irrespective of whether there is a single trial, there is funding for sites to invest in personnel at the site, and this includes the PI. Co-PIs at 20%, which is 1/5 of their time, is covered by the consortium and is expected to be an early career investigator. A site liaison, who is a member of the research team at 50% effort, and a community engagement specialist to build these partnerships with the community as a full 100% position. And there are separate RO1s that cover the grants. And the emphasis is on diversity and inclusiveness of study samples, innovative designs, central IRB, but most importantly, open data sample and image sharing. So for those of you who have been doing all these amazing analyses using the data sets here, you also have opportunities to work with clinical trials data as they are released. The A4 pre-randomization data is out. The full A4 data set will be available in a couple of months to everybody in the field. So I really encourage you to start looking at those data sets as well. This is what the consortium looks like. It's an extremely distributed national consortium with multiple institutions, 38 member sites, which is where the expertise around clinical trial conduct happens, 13 units with subject matter experts, several committees, a research participant advisory board, an internal ethics committee, and an external advisory board, really to get across the scope of the fact that this is a national program, very similar to the ADRCs. It's just focused on clinical trials. And we've taken an approach in this second grant cycle, which started about a few months ago, around inclusion and training as being a big part of what we do. And I'm talking about it from two perspectives. Inclusive trials through recruiting trial participants. And this is under the purview of the Recruitment, Engagement, and Retention Unit that I co-lead with Josh Grill. And workforce and leadership training, which is its own specific unit called the IDSCT, or the Inclusion, Diversity, and Education in AD Clinical Trial Unit that I co-lead with Risa Sperling. And I want to talk a little bit about our thinking around this, what we do, and then I will end with my favorite part of what I do, which is the Impact AD program. In the RER unit for all ACTC trials has defined successful trial participation as inclusive participation. And we think about it in three different dimensions. Race and ethnic underrepresented groups, which all of you know and are aware of. It's an important dimension to consider. Sexual and gender minorities as its own dimension. And then socially underserved groups. And we look at it in multiple different ways. We look at rural communities using what's called a RUCA score, look at scores between 4 and 10, high school education or less as a separate measure of socially underserved groups. We use a modified Hollingshead, which is a combination of education and occupation as a measure of socioeconomic status. And Dr. Amy Kind and her team here has developed the Area Deprivation Index, and we use that as a measure of disadvantaged neighborhoods. And all of these are collected for all ACTC studies through our standard minimal data set. So the idea is to standardize these critical elements so that we can evaluate it within a study and across studies. Yeah, I keep doing this. All right, I'll give you a minute. I keep doing this, sorry. But that's really what these underrepresented groups are: race and ethnicity, sexual and gender minorities, and socially underserved groups. I'm a statistician, and so for me, I need evidence based on data. And so that's one of the aspects of data collection that we have focused on. So if you look at this inverted funnel, it really is the recruitment funnel. At the top of the funnel, the first three segments of the funnel are things that are being done but are not being evaluated and captured in a manner that would allow evidence-based interpretation. And that includes the awareness, pre-screening activities, and eligibility. And so what we have been doing for the ACTC is to start capturing that particular data through what is happening in the pre-screening process. Are we reaching the people we need to reach? What approaches work for whom? What approaches don't work for whom? As you're going down this funnel, who are we dropping and leaving? Why? Can we make modifications to our inclusion and exclusion criteria? Does it impact the safety and efficacy of the study? If not, can we make these modifications to allow individuals to be enrolled in the study? So using data to allow us to evaluate what we're doing and how. So I want to talk a little bit about a couple of enrichment strategies that we've been using for the ACTC, and this is the AHEAD study again. It's using adaptive risk algorithms. So I'm sort of going all over the place, but adaptive risk algorithms uses machine learning models to be able to take the data and develop algorithms to identify individuals who may be amyloid positive. And if they are amyloid positive based on blood, they move forward in the screening process to the next cell. Else they get screen failed out and move into a longitudinal cohort called APEX. And this process continues, and once you get to PET, that's a definitive measure of amyloid levels. Those who have elevated levels are eligible for A45, those with intermediate levels are eligible for A3, and those on PET who don't have any of these levels then become eligible for the longitudinal cohort. So the idea is if you've made it all the way here, you want to find a way to keep these participants in the study, even if it is through a longitudinal evaluation, so that when the next trial comes along, they might actually be eligible for that particular trial. The second project that we're talking about is we want to go move into the community through the remote collection of plasma markers. And that's why the blood plasma has completely changed the field. We now can move into the community. And we do this through what we call the ALZmatch program, where we either through our online web study or directly through phlebotomists at these community events, we collect blood, send it to Quest Labs, send it to, in this case, C2N, The data comes back, we run our algorithm, and we link participants who are eligible with the appropriate clinical site. So again, moving away from sites at the pre-screening stage into the community to improve access. And so transitioning to the last part of it is around workforce development, education, training, and leadership, the IDRCT. The effort is to create an equitable and inclusive environment within our activities, to create networking, training, and education programs to support the staff, early and mid-career investigators, as well as emerging leaders. So it's not one or the other, it's actually both. And to create opportunities for the advancement of the next generation of ACTC leaders. One of the things that we felt very strongly that was important to do, it's not enough to train individuals, you've got to give them opportunities to become leaders and take over the leadership of the consortium over time. So we hope that in the next 5 years, should we be fortunate enough to get renewed again, the people leading these units and consortium are going to be very different people than who is there today. And we do this using multiple models, a leadership model where individuals, the early career investigators, are part of the group that is mentored around grant review or protocol review. And they had a formal vote in deciding which studies to actually be conducted at the consortium. So really bringing them into the scientific conversation, as well as having the site liaison of the research staff really help drive a lot of the improvements in site and study management. Engagement and networking, as well as research sessions, just like you had to share the work, except that it was focused on trials. I'm just going to skip through this, but there's been work that shows the need for this trials workforce. It's not something that anybody can just step in and do. You really do need to build an expertise around clinical trial design, conduct, and analysis. And this is something that the national plan addresses in ADRD as we talk about trying to find clinical therapies that can slow or prevent the onset of symptoms. AD trials are complex, they're multidisciplinary and unique, and training is required for that specific aspect. And there's a critical need to increase the pipeline of qualified investigators to design and conduct these trials. But equally critical is that the makeup of these pool of investigators be diversified. So the idea being that the investigators in your workforce has to look like the individuals that you are trying to enroll in your studies. So I'm going to end with probably my favorite week in my whole work is this 1 week where we all come together for the Impact AD course. Josh calls it a clinical trials boot camp. We spend 1 week together, faculty and trainees, and all we do is learn and talk about trials. Probably my favorite week in my professional life. And the Impact AD program is a comprehensive interactive modern training program to provide the skills and training for promising investigators who are interested in moving into or taking their careers in ADRD clinical trials research to the next level. So we look for individuals who are either trialists in other therapeutic areas who want to come to Alzheimer's disease, or individuals in basic science, translational science, who are interested in moving into clinical or observational studies, interested in moving into clinical trials. We leverage the collective expertise of the ACTC, and this is a program that's funded by NIA and the Alzheimer's Association. The objectives is really that facilitate career choices towards ADRD clinical trials research, to increase the pipeline, and to position trainees to secure grants to allow them to conduct their own trials. You have 2 tracks, the professionals track, which is for anyone who's a clinical trials professional, and this is at any stage in career, research staff or investigators interested in furthering knowledge and advancing careers in trials, and then the fellowship track, who are the future PIs of trials. The 2 tracks are different only in that the fellowship track has a protocol, and these protocols are reviewed by protocol working groups that include at least 2 clinical and 1 biostatistics faculty, but 4 trainees to 4 faculty. The curriculum is pretty comprehensive, both didactic and interactive sessions, but really focuses on elements that we believe are important for ADRD trials research. You can get the rest of the training everywhere else, CTSAs, stats programs, biostats programs, medical school, But what we believe are some elements around ADRD research that requires unique training, whether it is study design, whether it's statistical methods, outcomes, cognitive, functional outcomes, new gen outcomes like digital, as well as biomarker, biofluids and imaging, recruitment retention science with dyads as our recruitment unit, ethics, including ethics of disclosure. So lots to talk about in ADRD trials. And here's some of the data to show how we've been doing so far. And this is looking at our cohort for the last 4 years. And as you can see, we want to make sure that there's no bias in who we select compared to who apply. And we also want to make sure that we have a group that is inclusive. And so this is just some data around some of the key metrics that we are evaluating. But in summary, what this translates to is that we've had this for 4 years with 151 scholars, 78 unique institutions in the US, 35 unique states, 25%, 1/4 of them are first generation college, 53%, over half are from underrepresented racial and ethnic groups, 50% are individuals from institutions outside of the ACTC network, and 70% are women, sort of what the cohort is, the alumni scholar cohort is right now. This past year, we have gone global with the support of the Alzheimer's Association and in collaboration with the Global Brain Health Institute's Atlantic Fellow Equity in Brain Health program. We now have a global pilot program for Impact AD. This past year, we have individuals from 4 countries as part of the fellowship track, from Argentina, Brazil, South Korea, and Spain. And we have a lot of interest in this track this year as well. So it'll be interesting to see which countries, who joins this track, again, as we start building the clinical trials expertise globally. And that is when that's where the course is. And then if anyone's interested, you can still apply. It's due by the end of this month. So really hope that if I can get 1% of you to be at least just consider a career in trials, I think I'm good. That's really what I was hoping to accomplish this today. So if you're interested, please check us out. If not, just find me anytime. And so I'm going to quickly end talking about accelerating progress. So how do I think we need to move forward? And what are the areas that I believe are really most important to focus on? The first is we have to continue to design and conduct internally valid explanatory trials. We have to find new treatments that are safe and effective, and they have to be internally valid. You can't have external validity without internal validity. So that has to be a primary goal. But we have to move as close to external validity as possible and keep evaluating whether our criteria, whether our study designs allows the maximum access to individuals who might be interested in being a part of our trials. We have to invest in the training of the next generation of investigators and workforce, and not just training them, but also promoting individuals to leadership roles to help design and lead the next generation of trials in this space. To invest in infrastructure and partnerships through national and international clinical trials networks that include lower and middle income countries. Is something that I feel like is part of my long-term vision, personal long-term vision, is really to build these global trial networks to conduct clinical trials, farm trials globally. We're still not there, have a long way to go, but we're taking some first steps, but we also have to make it as national as possible. And finally, hopefully for all of you who are thinking about research and research in trials, to expand opportunities for innovation and collaboration across networks, like across ADRCs, ACTC, and programs with a focus on trial design and conduct. And I think there are many ways in which we can do it. And here's where I put all my hats together and say statistical methodology is the first. AI is everywhere. Artificial intelligence is everywhere. It's important to know what its strengths are. It's important to know what its limitations are. An estimation model is very different from a prediction model. Your problem drives your model, not the other way around. And so we're using a lot of prediction models. We need to make sure that these prediction models are not biased. We don't have algorithm bias in these models. These are things that us as researchers have to keep thinking about. So there's a lot of room in this space for more research and collaboration. we have to start thinking, and I know we've been thinking about next-gen outcomes, whether they're clinical outcomes, functional outcomes, biomarker outcomes. Got to start thinking about ways in which to make our trials simpler. Imaging, biofluid, and digital outcomes are really what we are seeing as the next-gen outcomes in this field. I'm a big believer that recruitment and retention is a key element in what we do, and we have to start building an evidence base around recruitment and retention science. What works, for whom, why not, and most importantly, what does not work, for whom? Does social media work better in some groups? Does it work better in some regions of the country? Is earned media better for in some parts of the country? Is it work? So those are research questions that you can actually use an evidence-based framework to ask and answer. And so integrating recruitment trials, integrating recruitment studies into the screening and the randomization process, I think is really important. And finally, we've got to get closer to the community and increase access. Without increasing access, there is no concept of inclusion. And so improving access through community-focused approaches to participant engagement and selection, especially during pre-screening. So removing the burden of pre-screening a little bit away from the study sites and the study personnel towards more centralized efforts, working directly with community partners, working directly with community resources to be able to identify individuals who are interested in trials, who have the biological elements around participation in trials, and then connecting them with the study sites to talk about a specific trial is really the way we see the future of the pre-screening process. As these biomarkers become better and better, blood markers become better and better, I think we have a great opportunity to start moving closer and closer to the community. So nothing that I do is by myself. We work together as a great team. Many of you here in the room, but I just wanted to acknowledge the team that I work closest with, and that is my team at the Alzheimer's Therapeutic Research Institute, including the biostatistics and the recruitment, engagement, and retention teams, and my collaborators, both here and outside, as well as our ACTC steering committee. Nothing in trials is done by one person, one group. It's a team effort, and it is the best of team science, in my opinion. So with that, I want to stop and thank you for your time. I also want to apologize for the fact that this wasn't moving in the same direction as this one. So thank you for your patience with this, and thank you for your time.
Amazing. Oh, you realized what you were, you really got what we said you will get. Now we have time for 2 questions. Rema.
Yes.
Oh, the mic will come out. Let's hear.
We have an online question.
Yeah.
For the demographics, would Indigenous scholars be under other, or are there none to report?
That's a great question. Right now, there are none to report, but I hope we can change it soon. And it would not be under other. It would be under the respective category.
Mm-hmm. Any more questions? So now, if one wants to take part in your course, how is it open to all of the world now? And
yeah, that's a great question. So the course in the US requires, if you're in the professional tracks, it requires your own letter of support from your team, from somebody in your mentorship team, plus your own indication of interest with and your bio sketch. If you want to apply for the fellowship track, you require a protocol as well as something that is being developed. You need to add that as well. The international track, the global track, we ask that anybody who's interested communicate with Dr. Heather Schneider from the Alzheimer's Association, and we will be reviewing it through the association's leadership. So, and again, sorry I don't have it's REMAR is my email at usc.edu. So just feel free, if there's any questions that any of you have, please just reach out directly, totally reachable, or ask anybody here. They know how to find me, or Google search me. I'm there. Way too easy to find sometimes. but just find me. But really, I just hope all of you recognize what a momentous time we are in this field. And I'm just so excited to be here and hear about all of this research, because it's only jointly that we're going to solve this problem.
So now it is my pleasure to invite the Vice Chair for Education and Faculty Development, leader of the Public Health Pillar at the Wisconsin Alzheimer's Institute, and co-leader of the ADRC Outreach, Recruitment, and Engagement Core, Dr. Art Walaszek, who will introduce our next featured speaker.
I have one job, which is to not butcher Spanish names. So I'm gonna try. And I want to say this is what a great testament to the power of being a part of this community doing Alzheimer's work. I'm really proud to be here and proud to have this opportunity to introduce my colleague, Dr. Maria Mora Pinzon. So Dr. Mora Pinzon is a physician scientist who is board certified in preventive medicine and public health. She graduated from the Universidad Central de Venezuela Escuela Jose Maria Vargas in Caracas, Venezuela. How was that? All right. And completed her preventive medicine residency at the University of Wisconsin, Madison. She is currently an assistant professor on the tenure track at the University of Wisconsin, Madison, Department of Medicine, Division of Geriatrics and Gerontology. Her work seeks to improve healthcare access for patients living with dementia and their caregivers, particularly among Latino populations. Among her many awards, she was selected as 2018 Young Physician of the Year by the National Hispanic Medical Association and 2020 William Kane Rising Star Award from the American College of Preventive Medicine. Dr. Mora Pinzon is a REC scholar from the ADRC and received a K99 ROO Career Development Award from the NIA to fund her project Improving Access to Alzheimer's Disease and Related Dementia Care Services for Latinx individuals at community health clinics. So without further ado, Dr. Maria Mora Pinzon.
Thank you. Okay, so thank you everybody for being here. And I know we are at the end of the day, but it should be fun. So the title, What Latinos Think About Alzheimer's Disease and Related Dementias, an implication, a population survey and implications for future research. Let's start with a couple of disclosures that you already heard. I have received funding from NIA, the ADRC, and also ICTER, and who else? And well, the main thing today, we're going to show you some of the results, preliminary results for my K99. But the first we're going to split this in like 4 different sections. First, what do I mean by the term Latino, Latina, Latine, or Latinx? Methods, preliminary results, and implications for future research. We know that Alzheimer's disease affects many underrepresented communities, many racial and ethnic groups. When I started working in Alzheimer's in 2017, I thought, yeah, Latinos, we don't get that. We're good. Lo and behold, we are 1.0 times more likely to develop Alzheimer's disease, and we expect that by 2060, the number of Latinos with Alzheimer's disease and related dementias will increase by 900%. If we don't prepare for that now, we are not going to be ready to treat them. But then who is Hispanic or Latino? According to the census, Hispanic can be viewed as heritage, nationality, lineage, or country of birth of the person or their parents. And people can identify as a Hispanic, Latino, or Spanish, may be of any race. This might be changing soon with the new census categories, how they're going to put it in the next few years. But the main thing is that there's not one single term that we can use, and you'll see why. So Hispanic refers to people that speak Spanish. That's, I think, the summary. And this includes people from Spain and any other countries where Spanish is the main language. Latinos refer to everybody that's from Latin America, whose lineage comes from Latin America. The main thing I want to highlight is that then, for example, using the word Hispanic leave people from Brazil out of most of our statistics. And even for census purposes, Brazilians are not included in the category Hispanics or Latinos. They can only be seen under white. But now you might be saying, okay, I know that. What's the difference between Latino, Latina, Latinx, Latinx? Which one do I use? The short answer, it depends. The long answer, it depends where you are. There's been multiple news articles saying Latinx is not something that we use. It doesn't represent the community. There's more recently been a push saying that's Anglo. We don't use that sound, the X at the end in Spanish, so it's grammatically incorrect. And there's even more push saying this is academics trying to impose a label on us. But again, this is very generational and very location specific. Here in Madison, we use in community events the word Latinx without issues, because it's been for many years something that we have used to reflect that it's not just female and male, that there's a range in between, and to acknowledge non-binary and all of members of the LGBTQ community that might be in this discovery process. So more recently, one way that we can see it is like, okay, we can use Latinx or Latine using the E at the end as a way that really reflects the Spanish language. Whichever word you use, it would really need to depend on the community. For example, for my working with in Milwaukee with the seniors, they say, we are Hispanic. We are not Latinos. We'd rather they use the word Hispanic. So many of my surveys, you will see multiple terms used, including Hispanic, Latino, Latine, Latinx in different places. But what do people prefer? We know that people prefer the country of origin. If you ask people what's the preferred word, it's usually gonna be the country of origin. I'm Venezuelan, I'm Colombian, I'm Guatemalan. That will be the preference. If you make them to choose between Hispanic or Latino, 50% will say no preference. That preference then will come down generationally speaking. Whatever you use, be prepared. We got this response a month ago from my survey. My survey included 5 different terms in different places. The word Latinx was once. This is the email that we got. I'm Hispanic. Latinx doesn't exist in the Spanish language. No one in my family uses the word Latinx. I'm offended. That's a strong word for me. So we adjusted the language just to make sure we deleted that, because we don't know how many people else feel that way. This is a national survey. Madison, we're good. But then how many people is not feeling like up to filling out the survey because they're offended? Just food for thought. But the methods that I'm going to describe today is that survey. So this is a validated survey that we started a few years ago, thanks to funding for the Department of Medicine, the pilot program, but also some funding from the ADRC. It has 47 items. It's available in English and Spanish. It's distributed online, in person, at community events, and it has been distributed so far in 6 cities, actually: Chicago, Puerto Rico, Houston, Wisconsin, Los Angeles, and New York. We're really trying to hit multiple representations because the way the Latinos are distributed in the US, it really changes where you are. And one of the things that I would like to highlight, the online survey, you can switch between languages into English and Spanish and vice versa at any point, and that took a lot of effort. But also all of our materials are available, everybody received them bilingually. So if you get a flyer, the flyer is in Spanish in one side, in English in the other, and with multiple options to fill out the survey. One of the issues I want to highlight, the online surveys, so anybody that has done an online survey know how hard it is this day with the bots and everything else. We have at least 6 different methods to catch bots and security measures, including the CAPTCHA, exact sentence replication, where basically we ask somebody, could you copy this text? If it's a bot filling out the survey, there will be gibberish over there. If it's somebody that's an actual person filling out the survey, they will fill it out correctly. Fraudulence called for Qualtrics. These measures based on your computer, IP address, the platform that you're using, they identify any red flags like, oh, this person already filled out this 10 times, so this might be a fraudulent response. Mismatch between name and email. Of course, if you see John Smith in the name and then the email is 12345 JA11 at Gmail. Yeah, that's not probably accurate. And we have data that support each of these steps. Response time, when we see the survey have been completed in 1 minute, 47 items in 1 minute, yeah, that's not feasible. So those are other ones that we are flagging, and abnormal behavior. This one, for example, when we saw the 5 responses being completed at the same time, that's a red flag. And we had to look a lot into, is this has been described before? Because I never heard about that. It's like, well, what are the chances that 5 people complete the survey at the exact time? So these are our security measures just to make sure that we have high quality data. But now let's see the data. So far, we have 591 Latinos in the United States that have completed the survey. And I have 200 more surveys that are in paper that we are inputted into the system, and we are closing recruitment hopefully next week. So this number will go up. 26% of them are men, which I think is great because they never want to fill out surveys. No offense. 1% non-binary, 73% women, but we have seen this in many research studies. And I'm really proud of this distribution by country of origin. Again, one of the main things that we try to make sure is that we really get different representation for all the different countries of origin. So far, 42% of our sample is Mexican American. They represent about 70% of Latinos in the US. So we are really seeing a lot of overrepresentation of the other groups. 18% Puerto Rican, 13% from South America, 18% other. Here's a detail. When we ask them, what's your country of origin or lineage? Many just write Latino or Hispanic. So those are the ones that we have in other or missing, because everybody that gave a country, they are represented in these other ones. And the language, I want to highlight that these really match who are you trying to recruit. Older generations, first generation immigrants are more likely to read news and use Spanish language in most of their life. But second generation and above, English is the preferred language. So thinking about implications for research, who are you trying to recruit? If you're trying to go with the older groups, Spanish is more feasible, but if you are okay or your research includes a lot of younger or middle-aged caregiving partners, English is usually the preferred language. What is the results of the survey? 74% of people think that forget thing is just normal. Good news is that only 38% of them thought that Alzheimer was normal. And then another study that I'm not reflecting the results here, the use of words Alzheimer's, dementia, memory problems in the community is very hard to track. Most of the people believe that it's a cycle where you start having memory problems that develop into dementia, that develop into Alzheimer's. That's the perception based on our interviews before. 75% of the people think that you can do things to reduce your risk of Alzheimer's disease, and we'll talk about that in a second. And about 70 to 80% correctly identify what are some of the signs or symptoms of Alzheimer's disease, like forgetting appointments, having difficulty finding words. So there was a very good information here of that it is not an issue of knowledge. There's very good knowledge around. When we go deeper into the risk factors, most people, 86% adequately refer that one of the main risk factors for Alzheimer's is having a family member, a parent that had it. Good. Then when we go down the list of poor sleep, stroke, nutrition, TBI, hypertension, diabetes, you see how this is going down. But interestingly, even though only 54% thought that hypertension was part of the things that might increase the risk, 85% of people thought that by controlling hypertension, you will improve your risk. So there's a very good, in the Latino community, what we're seeing is that there's a high level of perception that what happens to our bodies is a result of how much we take of it in our earlier years. So very good reception of all of these lifestyle changes in general. And this is, if you want to take one thing out of this talk today, we ask, how important is that people with Alzheimer's disease be cared for at home by family members? 74% said that was very or extremely important. 19% somewhat important, and 7% little to no important. If you're doing research on nursing homes, long-term care facilities, or any other institutional populations, and you come asking me, where are the Latinos? Yeah, they're not there. They're at home. And this is one of the things that my research tried to emphasize. Is this an issue of access or is this an issue of preference? It is an issue of preference. So how can we empower and provide the tools to families to take care of their loved ones at home and decrease the burden? Because most of the interviews that we have heard, for example, there was one that said, the doctor told me that I needed to send my mom to a nursing home. We never went back. And I always go back to that quote because how many people are we missing, not only for care, but also for research, because they lost trust in their medical providers? Which leads me to the next point about the trust. According to the behavioral risk factor surveys, only about 41% of Latinos will talk about memory or cognitive decline with their healthcare professional. Question for me was, is this because they think it's not important, or is this something else? It is something else. Because 88% of the people in my survey would like to talk about their memory problems if they were having them with a doctor. 92% of them thought that the doctor could help them in some way. But 57% of them will find it embarrassing. And this is a population that saw the survey, and the survey title is, we want to know what Latinos think of memory problems, Alzheimer's, and dementia. So we know that they're engaged. We know that they have some knowledge or at least interested in the topic. Still, 57% of them were like, I don't know how I'm gonna do this part. And 50% will find it easy to ask for help. This was a very reassuring number because that means that at least 50% of them knows who to ask. How are the implications of this for the future research? Well, first we need to distribute these results. One of the main things that we have heard in the process of recruitment is that people say, what are you going to do with this data? And people want to know the results. They want to see the infographics. They want to see something that gets to them and not just a nice paper in a journal. They want to hear about what we learn and how we're using that information. So that's part of a lot of the outreach that we do. Analyzed based on country of origins, 52% of the sample has at least a college degree. So that's higher than the average population in the United States. So right now, one of the next steps when we have the full data is to identify, are there differences in knowledge and perceptions based on your level of education or even country of origin? Are there messages that will resonate more with Mexican Americans or Puerto Rican Americans, or what is the distribution that we will have? And finally, and the main thing, healthcare professionals have a high level of trust. 92% of people thought that a doctor could help them if they had a memory problem. I bet that if we ask the question to a doctor, it will be a very different number. But the main thing is that if we can leverage, and I think this goes to Dr. Raman's presentation earlier, we can leverage healthcare professionals in the community to participate in research in some way or form, we can probably get more people interested in research. There's a lot of good knowledge and interest out there. And we got many responses after the survey say, hey, I love this. I love the work that's happening. If there's any other studies that I could participate, please let me know. So there's a lot of interest. It's just about leveraging our tools to make it happen. With that, thank you so much. And thank you guys for having me.
That was a wonderful presentation. We have time for a couple of questions. Please raise your hand and we'll bring you the mic. Or if you are on Zoom, please feel free to drop a question in the chat.
I'm curious about the 18% of people who just wrote Hispanic or Latino. And I'm wondering if those individuals didn't want to disclose or if they had like too many countries of origin.
Great question. So people could actually select multiple countries in the survey. So some of them are already included in the other numbers. But what we have heard is just comes down the lack, they don't want to disclose, or there's also a fear of disclosure that they don't know. Partially in our survey, we need to ask them name and email to give them their thank you for their participation. And in those cases, they say, yeah, I don't want anybody to have any idea where I'm coming from. So that might be one of the things playing a part here.
My question is, how many families who are caring for the Alzheimer's individuals at home live in extended family units or have multiple people contributing to that effort?
Yes, according to statistics, we have seen that multi-generational households in the US in general for Latinos, it could go up to 60% depending of the region. One of the last studies that we saw is that about 40 to 45% of people with Alzheimer's disease are cared for in multi-generational households. Well, those are national statistics. I don't know if this truly represents this group, I want to say, but we asked how many of them were caring for somebody, and at least 60% of them had at least 40% of them had experience of caring for somebody with Alzheimer's disease and dementia, but we did not ask about the multi-generational household.
All right. Thank you, Dr. Mora Pinzon, for such a wonderful and informative talk. All right, we are now going to turn to the award session. I'd like to start off by thanking Dr. Sanjay Asthana and the Asthana Family Fund for supporting the Alzheimer's disease and related dementias Research Day poster session and lightning presenters. And I'd like to invite Dr. Asthana and Dr. Johnson to please come up to the stage to help me in recognizing the winners for the poster session and the lightning presenter awards. All right, when your name is called, you'll please come up to the stage and receive your award from Dr. Johnson and a handshake from Dr. Asthana and money from Dr. Asthana. So, all right, our first poster award winner in the undergraduate category is Lauren Gitzlaff. Lauren, do you want to tell us what the topic of your research was?
Yes, so I looked into brain microstructural changes in relation to rock stage progression.
Congratulations, Lauren. And please stay on stage because there will be photos. Our next winner in the graduate student category or research specialist is Abigail Everett. You can tell us what your research is and then receive your award.
Awesome. Hi, everyone. I'm Abby. I study NLRP3 inflammasome activation in macrophages and microglia. Yeah.
Congratulations. And the next award in the fellow postdoc or research scientist category goes to Tamara LeCaire.
Remember the introvert comment earlier. I am working with Art Walaszek and Cindy Carlson at the Wisconsin Alzheimer's Institute and Maria. And we, the poster was an implementation methods qualitative research poster for our academic detailing and DICE model for addressing BPSD in individuals and clinician education for that.
Thank you for that great work, Tammy. All right, and now we are going to have our fan favorite poster awardee and our fan favorite lightning presentation. So the fan favorite poster presentation is actually from a prior award winner, Reggie Baby Giridja. Please come up. Thank you. Oh, okay. Absolutely. So we, Reggie sent a representative to pick up her award. But you could also introduce yourself to us, please.
I'm Michelle Sensala. I worked with Reggie on her poster. It was on protein restriction and how it may be beneficial in Alzheimer's disease using the 3xTG mouse model.
Excellent. Thank you very much. And now the fan favorite lightning presentation. They were all fantastic, first of all. But the award goes to Rachel Audie. And Rachel, we saw your talk, so you can just take your word. I won't make you repeat yourself. All right, let's get a picture of all of the winners with Dr. Astana and Dr. Johnson. All right, wonderful, thank you. And I just wanted to ask all of the winners to also see Cheryl when we close out because she has something for you. So I just want to thank everybody again for their posters, for their presentations, for their engagement, for all of you being here today, including Clint. Thanks for coming, Clint, and taking photos. Yes, Clint. We really couldn't have done this without all of your engagement. Thank you again to Cheryl Jean Spenceley for all of your organization. Wonderful work, keeping us going. Thank you again to the Discovery Center and helping us with IT, keeping us on track. All of the poster judges, all of the volunteers, all of the faculty that came here today to support the scientists and all of our speakers. It really was a very special event, and we heard about so many different topics today, mechanisms of Alzheimer's disease and etiology, treatments, modifiable risk factors, social determinants of health, biomarker innovation. We had a lot of biomarker talks, so go biomarker teams. And, as well as, of course, what we've heard about inclusion and ensuring that we do the best science that serves people who are affected by Alzheimer's disease and related dementias. And we also had a great day of collaboration, connecting with friends. So thank you all for joining us. What we are going to do now is we actually are going to have the opportunity to have a bit of a social out in the open court. If you have your ticket, that is good for one wine, beer, or soda. If you don't have a ticket, I have a literal Tupperware full of tickets right here. So that's if you're missing your ticket. We do want to be able to do this again next year. And just some other comments before you leave today. Remove your name tags, place them in the bins on the registration table. Remember to take your posters. And thank you to everyone who helped make ADRD Research Day 2024 possible. Thank you for joining us.